So now what we want to do is talk about reproduction in bacteria. That's certainly part of growth and nutrition, is the ability to reproduce. Bacteria undergo asexual reproduction, which means that they do not need a partner in order to be able to reproduce. They can simply divide in half. And there are other organisms that can undergo asexual reproduction. If you've ever had a spider plant, you've probably seen it grow these little plantlets that come off, and you can actually separate them from the mother plant and plant them and have a whole new plant. You can have a house full of spider plants in a few years. So anyway, this spider plant is looking at a dating app, and he says, I don't have time for this. And I'm sure all of us can relate. Anyway, bacteria, like spider plants, undergo asexual reproduction. In bacteria, we refer to this process as something called binary fission. And you're going to be really happy to know it's super simple. So we have a mother cell here, and the mother cell has inside of it lots of things, but we're focusing in this diagram on the ribosomes, plasmid DNA. Those are those little extra circles. Some bacteria have them, some don't. And then the chromosomal DNA. And the first step is for the bacteria to divide the contents. So you'll have new ribosomes, you'll have division of plasmid, and you'll have copies of the chromosomes. So you need 2 copies, one for each of the new daughter cells that's going to come out of this division. The cell will elongate, and you'll notice that the genomes and contents migrate to opposite ends of the long dividing cell. And then what will happen is the plasma membrane will pinch off, and the cell will lay down new cell wall in between the 2 daughter cells. Eventually, the 2 daughter cells will separate, and you'll have 2 separate cells. And of course, if conditions are good, these bacteria will immediately begin dividing again, which is one of the reasons this process is so fast. This picture here is a gorgeous picture of a bacterium called Bacillus licheniformis. And Bacillus licheniformis, you don't think you're familiar with it, but if you wash your clothes with detergent, and if your detergent says contains protein stain fighting power, it probably contains enzymes made by Bacillus licheniformis that break down proteins. These are proteases, and they've been used extensively in genetic engineering to make detergents that are used in washing machines more efficient. Bacillus licheniformis, the enzymes, it's a bacteria that likes living in warmer temperatures, and so it can tolerate hot water in the washing machine. This is a wonderful little video. I'm just going to turn the sound off. This is a wonderful little video just showing bacterial growth. It's a time lapse, but you get the sense of sort of how quickly, if this is happening every 20 minutes, just how quickly you'll get a number of bacteria in a culture. It just does not take long to go from 1 to having a very thick culture of bacteria. And this leads me to talk about something called logarithmic growth. And when you are dealing with something that you start with 1 and it goes to 2, and then when those 2 cells divide, you don't go to 3, you go to 4. And then when those 4 cells divide, suddenly you have 8. We call this logarithmic growth because it's not growing 1, 2, 3, 4, 5, but it's growing 1, 2, 4, 8, 16, 32, 64, etc. And so you can end up starting with 1 bacteria, which is what this chart is showing here. And in just 20 generations, you can have 1 million bacteria. So that's why, in the lab, we can culture bacteria, and then you can come in really the next day, and you have a colony that you can see from something that was invisible. So bacteria reproduce incredibly rapidly. And that's shown in this cartoon by the mother cell here feeling overwhelmed at all of the offspring she suddenly produced. So kind of a cute cartoon from the Amoeba Sisters. They actually make videos, scientific videos that are quite good. And if we haven't seen any yet, you certainly will over the course of the semester. When you plot rapid growth, like the kind we're talking about on graph paper, what you end up seeing is, so the scale here on the right-hand side is a linear scale. So here's 1 million, here's 10,000. And what you see is it looks very flat in the beginning, but as you start to get bigger numbers, we end up going really quickly from 200,000 to 500,000 to 1 million. So the last 3 generations, you see this very steep increase in the curve. And you end up not being able to see all of the generations that have occurred prior very well. And a lot of times scientists will plot this kind of data on semi-log paper. So that's looking at a linear scale on one side and a logarithmic scale on the other. So here, instead of a linear scale, we go one is, when we go from 1 to 2, this is actually a 10-fold increase. So from 1 to 3 is a 100-fold increase, and this is 1,000 times more. And when you plot this on semi-log paper, you get a straight line when growth is logarithmic in this particular case, or exponential. And this is helpful for bacteria because you lose a lot of information on a linear curve. More importantly, especially if math isn't your favorite thing, I want to go to the next slide and show you how we use this when we're studying bacteria. So this is the same kind of paper. This is our log paper. So it's a log scale on one side. That means that every step is 10 times more than the prior step. And then a regular linear scale on this side. And when you plot growth, what you see is bacteria and culture have a very characteristic pattern. They begin by starting something called lag phase. And in lag phase, it's a little bit like building a house. Before the house starts to go up, you have to get all the supplies to the building site. And so in lag phase, nothing seems to be happening, but in reality, the bacteria are preparing for a big increase in cell division. Once they get going, we head into log phase. And so this is where the generation time, we're going from 2 to 4 and 4 to 8 and 8 to 16, and we're just getting an extreme exponential increase in the number of bacteria that are in the culture. At some point, the bacteria start to die off, and you have some bacteria dividing, but some bacteria dying, and so you end up with a balance. This is a period of equilibrium where the number of bacteria that are being produced are equal to the number of bacteria that are dying. And then we have death phase. And so in death phase, what we see is that now maybe the nutrients are running out, and what's happening is the population is beginning to decrease. We have more death than we have bacterial division. This is really important. It's something I'm going to be bringing up in the lab frequently, and that is that you want to keep your cells in log phase and stationary phase to ensure that you have plenty of healthy cells for subculturing. And when we talk about this during the unknown project, I'll be reminding you pretty regularly to keep your unknowns in log phase where they'll stay healthy and grow for you the way you want them to. Okay, so that's it for binary fission. Our next topic is going to be spores, because some bacteria have a special capability of shutting themselves down and saving themselves when conditions are rough and waiting it out until they get better again.